Hello friends, welcome to BMH Learning. This lecture is about protein targeting to lysosomes. This process will involve mainly two organelles, the Golgi apparatus and the lysosome. So, first let's remind the outline of Golgi structure. The Golgi is formed of three parts. The first is cis Golgi. The second component is medial Golgi. And the third one, TransGolgi. Now, this cis Golgi is referred as receiving site. It's tasked with receiving proteins, mainly from endoplasmic reticulum. On the other hand, the TransGolgi is referred as shipping site, because from here the proteins are shipped or simply sent to other destinations of the cell. In this case, it's lysosome. Now, all the proteins which are coming from ER, they possess a preformed N-linked oligosaccharide precursor which is formed of 8 mannose and 2 N-acetyl glucosamine units. If you want to know how this N-linked precursor is formed, check the link in the description. So, we have one such protein which is delivered from the ER and we call it as lysosomal hydrolase enzyme. Now, to make it more simple, we will not include the whole N-linked oligosaccharide precursor in this protein. We will only focus on this mannose residue. So, initially the protein moves to the cis Golgi. Now, in order to differentiate this lysosomal enzyme from other ER proteins which are also present in Golgi, it undergoes a special treatment in the cis Golgi which adds a phosphate group at the 6th position of mannose residue that makes it the protein with mannose 6-phosphate. And from here, the protein shifts to the medial Golgi and then to the TransGolgi. Now let's discuss the enzymatic formation of mannose 6-phosphate in cis Golgi. So, the addition of the phosphate to the mannose is a two-step process. First step is transfer of phosphorylated N-acetyl glucosamine group to the protein. So, this is our lysosomal protein which is joined by N-acetyl glucosamine residue phosphorylated with the help of UDP. This reaction is catalyzed by the enzyme N-acetyl glucosamine phosphotransferase, which transfers a phosphorylated N-acetyl glucosamine residue at the 6th position of mannose unit present on the protein. Now the second step is removal of the N-acetyl glucosamine residue. So we have this modified protein now which is acted upon by second enzyme known as phosphodiesterase. This enzyme removes the N-acetyl glucosamine group and produces the desired phosphorylated mannose residue, which is ready to direct the lysosomal protein to its destination, the lysosome. So, this is the TransGolgi and our protein of interest is right here. For the transport of the protein, a bud starts forming, and the formation of the bud requires the assistance of two other proteins. First is AP complexes assemble around forming bud, and then the bud is surrounded by cord proteins known as clathrin. Now, the membrane of the TransGolgi has receptors which are known as mannose 6 receptors. So our protein binds to this receptor, and then receptor guides the protein to the bud. Now everything is in proper position, so the bud is pinched off and we get a fully formed clathrin coated transport vesicle. Now, it's time for the disassembly of the AP complexes and coat proteins so that vesicle can fuse with the endosome. So, we get a naked or uncoated transport vesicle which is now devoid of both AP and coat proteins. Now, the uncoated vesicle fuses with the endosome. So concentrate here fully because a lot of things are gonna happen in the endosome. As the protein encounters the environment of endosome, both the receptor and the protein are dissociated. This is because the receptor can bind to the protein at pH around 6.5, which is found in Golgi, and the pH of the endosome is slightly more acidic, which is maintained by the action of VATPase embedded into the endosome membrane. This proton pump continuously pumps H positive ions into the endosome, thus resulting in lowered pH of the endosome, and that's not suitable for the receptor protein binding. Furthermore, in order to eliminate any chances of rebinding, a phosphatase removes the phosphate attached to the mannose. So, what we end up with? This, the original protein form with which we started the mechanism. Finally, the protein goes to its main destination, the lysosome. So, the protein has completed its journey. Now what about the mannose 6-phosphate receptor? It says, it's not my home. My parents are waiting for me. So, it must be recycled back to Golgi for another similar pathway. So, to take the receptor back to its home, a vehicle is ready here. You can say it's a retrovesicle. Hence the receptor sits here and they both take off to reach Golgi. This was all. Thanks for watching.